session will work somewhat in terms of format similarly to the session that we had before lunch that Richard Barker so excellently chaired. I'm a little scared, really, because I thought that Richard did such a fantastic job that I've got to try to match it. But what we're going to try to do here, and this is going to be pretty much repeated tomorrow and on Saturday, is we're going to bring together speakers who have given talks in the concurrent sessions earlier in the day to not only summarize to the rest of you who weren't lucky enough to see their own talks what they said, but also to put it in context. First of all, to perhaps reflect on other talks that they've seen and say what their perspective on that context is. And also, after they've given their own remarks, to engage in discussion, first of all, among the five of us, and then, of course, bringing you guys in, concerning how this all fits together. You've noticed, if you've read the program, that in each day we have concurrent sessions, two of which are biomedical, covering different diseases or disabilities. So today it's been a thread on Alzheimer's disease and another one on cardiovascular. And then in parallel with that, a non-biomedical thread looking at aspects of the social, political, economic, demographic context of all of this. And in each of these panel discussions following those concurrent sessions, we will have participants from each of those threads. So in this occasion, we have two of the speakers from the Alzheimer's track, one speaker from cardio, and one speaker, Neil Lippman from CRM, who spoke in the non-biomedical track, so to speak. So we're going to start, as I mentioned, by doing that. And as Jerry mentioned, we have to curtail the session a little bit, but we made these sessions really long for a reason. We knew that, first of all, we had to have padding in the thing because everything always overruns. But also, the fact is, there seems to be a lot to say. I was extremely heartened this morning by how long the session ran and how difficult it was just to fit in everybody's questions. And I'm very much hoping that every one of these sessions will be just like that. So I'm going to proceed in the order in the program, which I can't remember. I'm going to start with Rochelle.
Shall I go here?
Go ahead, please.
Well, I realized that not everybody was in the same session, and I only realized that today. So what I thought I would do is give you a little flavor about my discussion, which was approaches to brain aging and Alzheimer's disease. What are we learning from clinical trials? And in order to talk about what we're learning from clinical trials, we have to be broad across different mechanisms of action and different strategies. I began with a discussion of the risk factors for Alzheimer's disease. In every population in the world, the driving force is aging, and the mechanisms by which aging promotes the risk are numerous. Menopause is permissive as well, but not an absolute cause. I reminded us that there are differences in our genetic backgrounds and listed a number of the genetic variations that confer risk for Alzheimer's disease: ApoE4, TOM40, CR1, CLU, PICOM, SORL1, TREM2, and made the point that really didn't originate with me, but with Peter St. George's Hislop. This genetic risk is probably cumulative. So although we may have gene variants that are rare, if we have several of them, our risk is probably higher than if we just have one. There are the middle-aged cardiovascular risk factors, which we overlap with the other panel: metabolic syndrome, elevated glucose, hyperinsulinemia, diabetes, cholesterol, elevated blood homocysteine. All of these, even in middle age, increase our risk of Alzheimer's disease later, and therefore highlight pathways, metabolic pathways in particular, and targets for potential intervention: major head injury with loss of consciousness, and chronic inflammation. I said that the pathologic factors, if we move away from this epidemiologic view and more to a tissue-based view of what happens in a disease, what happens in Alzheimer's disease, suggests another set of targets for different reasons. Plaques and tangles, which are necessary but not sufficient, suggest anti-amyloid and anti-tau therapies. The loss of cognitive reserve is the other side of the balance between the presence of plaques and tangles, which do not necessarily cause Alzheimer's disease, and the actual development of a disease. So what is it about cognitive reserve that perhaps is estimated by premorbid IQ, and how do we enhance that? And I didn't say it in my session, but I think we enhance it very early in development. And maybe in an aging conference, we have to start thinking about aging as starting at maybe age 6. So maybe we have to start enhancing cognitive reserve. Various aspects of the biochemistry of aging are targets, probably those things related to protein management and oxidative state as major targets, the genetics, as we said, and chronic inflammation. And the effector cell through which we are most likely to be able to modulate innate immunity, as far as I see it right now, is the microglial cell. So big epidemiologic view, molecular tissue pathology view, converge us on certain potential therapeutic approaches. The strategies for anti-dementia drug development, as I categorize them for purposes of convenience, would then be drugs and nutraceuticals. And today we heard someone ask a question, what happened to the nutraceuticals? What happened to vitamins? Well, they're there. They're there based on the epidemiologic observations. And so there are drugs and nutraceuticals based upon epidemiology that are potential treatments for Alzheimer's disease and potential prevention approaches. The neurotransmitter-based therapies, the drugs that are approved today are effective, and they hold up in multiple kinds of analyses, and they probably have effects that persist over the lifespan, despite what you may have heard. Neurotransmitter therapies remain a target. Glial modulating drugs, as I said, things that try to undo the effects of hyper-fed states, metabolic syndrome. So PPAR gamma agonists or sirtuins, these are metabolic neuroprotective approaches. And then if we can figure out exactly how to modify tau and exactly how to modify amyloid metabolism. So we went through some of those discussions. What have we learned from recent clinical trials that do not target amyloid? Okay, I was asked to speak about an amyloid target, but I wanted to put it in the context of these other studies. Well, we have learned that transmitter-based therapies continue to show clinical signals, and I mentioned several of them. And you should remember the alpha-7 nicotinic agonists and the 5-HT6 receptor antagonists among them. We learned that vitamin E has an effect in modifying course of disease. And we knew this from the early 1990s, one early trial. In our center, we continued to use this approach over the years and to study it, and we're not scared away by the possible increased risk of mortality for megadoses of vitamin E. And very recently, this vitamin E observation was replicated in a large multi-center VA-based study that high doses of vitamin E, 1,000 units twice a day, preserved functional ability in people with dementia. We've learned from recent clinical trials that when we choose a drug target from epidemiology, so menopause is permissive, what about estrogen? Non-steroidal anti-inflammatory drugs to target inflammation, B vitamins and folate to lower blood homocysteine, statins or DHA. When we choose our targets that way and give them to people with disease, they do not work. And that may be because it's too late. It may be because there are endophenotypes of disease, and when we group the people all together and give them these interventions, we can't see the effect. And then finally, we learned that placebo groups do not change the way they used to. I then targeted anti-amyloid strategies, the passive immunizations, the active monoclonal antibody vaccines, the anti-fibrillization and anti-aggregation approaches, and the beta and gamma secretases, and discussed the successes and failures among them. They represent not a test of the amyloid hypothesis, but a test of a particular anti-amyloid approach, one instance of that approach. Finally, I talked about recent monoclonal antibody trials, very large-scale studies with bapineuzumab, which is a monoclonal antibody directed against A-beta. It was characterized in its development as being an antibody that targets fibrillar A-beta as well as plaques. It targets the N-terminal domain, and it's associated with some vasogenic edema and microhemorrhages, especially in E4 positive individuals and especially at higher doses. Contrasting with solanezumab, which is another humanized anti-A-beta monoclonal antibody binding soluble A-beta only, mid-region antigenic target, central domain, less likely to produce vasogenic edema, and that was borne out in the studies. And then crenezumab, another antibody, this one built on an IgG4 framework to try to avoid overactivation of microglial cells. This binds monomeric, oligomeric, and fibrillar A-beta. Okay, so another difference, also a mid-central domain. In looking at the results of these three studies, the bapineuzumab had actually 2 large phase 3 trials, negative by its primary outcomes, but what did we learn? Well, we have claims based upon analysis, secondary analyses of secondary outcomes, that it may have lowered phospho-tau, and it may have resulted in less accumulation of A-beta. Yet this effect was not consistent using the same dose in studies of E4 carriers versus non-carriers. And it was not able to be generalized to the whole population because very, very, very small subsets of patients had CSF analyses or amyloid imaging. Solanezumab, I believe, again, my interpretation of the data, that there were very large excursions in A-beta in the plasma, and but the next speaker, Dr. Siemers, who followed me, suggests that some of those subfractions of A-beta in the plasma indicate that you were breaking up plaques in the brain and moving broken plaques into the periphery. I think that may well be the case, and I do believe that there was a signal, particularly in the mild subgroup, which is in one case a post hoc, not a post hoc, but a secondary analysis. In the other case, the primary outcomes that were chosen did not show the effect, but additional outcomes that are looked at give you a matrix of results. And the matrix of results suggests to me that there is a cognitive effect of lowering A-beta by this mechanism. And finally, crenezumab, very recently reported. I stuck to the data that were presented at the AAIC, Alzheimer's Association International Congress in Copenhagen last month. Crenezumab also, as I said, binds monomeric A-beta, and I believe there was a signal there. It was very tough to be certain, given the powering of the studies, which were both very small, and given the data available. But again, I believe that based on clinical grounds, there was a signal in the mild patients. So in conclusion, what I have said and put out into the room for discussion is that from the monoclonal antibody studies, I might conclude that the overall effect, if it's definitely present, is small, that effect may take several months to become apparent. And when I say several months, I mean almost a year. That the effect size may be larger in mild patients, that it may be a function of targeting soluble A-beta and not fibrillar A-beta, and that this approach remains a viable target in Alzheimer's disease. We talked a little bit about the fact that some look at the same data and they conclude that we should not expect much of an effect in symptomatic patients, that we should abandon that idea and move entirely to prevention approaches with anti-A-beta therapies. And indeed, several of these are underway, making use of populations, known collected populations of people affected by familial AD autosomal dominant mutations, or people who are homozygous for E4. And these studies are underway, introducing various of the monoclonal antibodies very early. So that's a background. What does that mean for preventative medicine, for strategies to enhance negative senescence? Well, it may mean that at some point we will choose people among the population for various reasons, either reasons of behavior or reasons of genetics or some biological difference, and we will be introducing therapies in middle age or even in preclinical periods 20 to 30 years before disease is expected to develop, and that anti-amyloid approaches may be among this menu of things that we introduce. Thank you.
All right, Neil, please. This is probably not a natural order in which to do the speakers, but that's okay. Since we do have to compress things a little bit, I hope that you guys can stick to maybe 6 or 8 minutes, no longer. Thanks very much.
Sure. So first of all, Aubrey, thank you for inviting me to speak today. Happy to be here. For those of you who listened to my earlier talk, I'll try not to be too repetitive. I'll try and incorporate a few new ideas. But for those of you who didn't, I'm the Business Development Officer at the California Institute for Regenerative Medicine. So CIRM was established in 2004 with the passage of Proposition 71, which is the Research and Cures Act, which provides for $3 billion of funding to fund stem cell research and associated technologies in the state of California. So ultimately, we will probably be measured on delivering cures and therapies to patients. That's our goal. That's our mission. That's what we're working towards. And to that effect, by the end of 2014, we expect that our grantee organizations will have 10 INDs filed, and we'll be enrolling patients in 10 different clinical trials. Those clinical trials range from everything from type 1 diabetes. ViaCyte is a company that recently filed an IND. Capricor, Linda Marban is here as the CEO of Capricor. They're in phase 2 for heart failure indication. Asterias is another one of our grantees. You might have heard Mike West talk earlier from Biotime. They'll be filing an IND for spinal cord injury. So overall, we have a portfolio of over 90 translational programs that span various diseases. We're looking to move those forward into the clinic. I think it's important. One thing I talked a lot about earlier was the idea of de-risking these projects. So part of CIRM's mission is really to provide non-dilutive capital to de-risk these projects, both in the academic setting and in the company setting, to try and entice the private sector to come in to leverage the public dollars. And so I think we've had some successes. Capricor announced a partnership with Janssen on their program, which Linda could talk about a little later. Sangamo announced a deal with Biogen Adec for a preclinical development program for beta thalassemia. So I think one of the other topics I highlighted earlier was the shifting of the funding continuum. And what I mean by that is traditionally industry was getting involved post-phase 2 data, had to see proof of concept data in humans. I think we're seeing industry getting involved a lot earlier these days. CIRM has a variety of funding mechanisms to try and de-risk these types of novel approaches. We have what we call a strategic partnership award, which provides up to $10 million of funding to complete a phase 1 or phase 2 clinical trial. It requires a 1-to-1 match from the applicant. But the idea here is for industry to bring in novel technologies to access non-dilutive capital from CIRM and to try and entice industry to come in and actually fund some of these programs. So since this panel is dedicated to cardiovascular disease and Alzheimer's, I just want to highlight CIRM's portfolio in these areas. So we have 51 awards totaling over $180 million in our cardiovascular portfolio. Many of them are focused on iPS cells, disease modeling, so using iPS cells to screen for novel therapeutics. We also have a number of translational programs. So Joseph Wu at Stanford is one of our grantees who has a $20 million grant to advance human embryonic stem cells, which are differentiated into cardiomyocytes for end-stage heart failure. He also has an early translational award where he seeds his cells on actually a scaffold, and it's a patch that's actually a bioengineered patch to place on the heart, which has some bioengineering and structural components to actually help, which is a different approach from the typical cell therapy, which is just an injection. And then, of course, I mentioned Capricor earlier. In terms of our Alzheimer's portfolio, we have 17 awards totaling about $62 million. These are typically earlier stage awards than our cardiovascular programs. Our most advanced in this area is with Stem Cells, Inc. They will be filing an IND over the next 4 years, so we provide them $20 million of funding, and they're using a homologous approach. So they have neuronal stem cells that they're using for Alzheimer's disease. It's an allogeneic platform, so theoretically it's supposed to be scalable, kind of an off-the-shelf therapy, which is more in line with the traditional pharmaceutical business model. And so with that, I'd just like to say that CIRM funds novel tools and technologies. We don't only fund the therapeutics, but we fund the enabling technologies to enable the delivery of these types of therapeutics. And I know this question will come up, so I should just address it head on while I'm up here. How much funding do we have left? Well, we have about $1 billion of funding left at CIRM. So it's been announced publicly before that our funding should run out by about 2016, 2017. That's not the case. The $1 billion should last us roughly through 2020. So we're not coming up to a funding cliff as quickly as otherwise may have been stated. So just wanted to make that clear. So, thank you.
Linda.
Taking my timer with me so that I won't go over. I'm always motivated by food. Anybody that knows me knows that if there's food involved, I'm going to hurry. So thank you so much for inviting me here today. I have enjoyed this so much. Usually I have to spend my days at investor conferences, which have a much different flavor. And today, for the first time in a while, I get to sit back and be a scientist, and I heard so many fascinating things. And it stimulated a lot of thought for me and sort of the trajectory of my career. I started as an academic. I have a PhD in physiology. I studied mechanisms of excitation, contraction, coupling, which is how muscle contracts, and cardiac muscle. But I never really wanted to be an academic. I wanted to go out in those days to pharma and R&D and make a difference in the world and bring therapies to people. And it seemed such an easy goal, and yet it turned out to be such an incredibly long and challenging journey. And now, somewhere along the advent of my career, as I went from academia to startups in which I worked in gene therapy, just as gene therapy took its biggest hit ever, and then ultimately have moved from the incredibly easy field of gene therapy into the easier field of cell therapy, just being a glutton for punishment. It's given me a chance to think about translational medicine. And I think today we have sort of the perfect blend of those that are interested in taking therapies from bench to bedside. And so I want to highlight for a few minutes some of the things that have happened in the regenerative medicine space in terms of cell therapy for heart disease, because I think it's one of the areas that's most interesting. It's an incredibly crowded field. We haven't had an approved therapy from the regulatory bodies for 14 years for heart disease. Part of the reason for that is it's really hard to treat. Part of the reason for that is that it requires very large, very, very expensive clinical trials. And so it's a little bit harder to take something, from bench to bedside. But what happened is that everybody jumped into this space. So I don't know, I last count, how many companies and how many academic trials have been sponsored to study cell therapy for heart disease, but I'm going to say that I personally know of about 20. So there's probably many, many, many more than that. And so what happened is that the early funders who came in excited about cell therapy for heart disease looked at early data that perhaps was not at all relevant to the end clinical endpoint, but something that they could sink their teeth into, ejection fraction, and said immediately, cell therapy for heart disease doesn't work. And this was in 2003 and 2004 with the REPAIR AMI studies. Now what's interesting, let's use that as an example of how a field can get clouded, but actually have something very good in it. Interestingly, while they just saw in that study that there was a 3% improvement in ejection fraction, which is modest and everybody said doesn't really matter, what they forgot to tell you, because they didn't know at the time, and now we know 10 years later, is that those people have an incredibly improved survival benefit from those cells. So that's something that we know about in science, but the general public doesn't hear very much about. And so we continue to move the field forward on actually what's really good and compelling clinical data, but a field that's so crowded and has had a history of getting funding that's not relevant to really move the field forward until, thank goodness, we finally have a pharma partnership. Maybe that'll begin to make a difference with J&J. And then we have CIRM, the California Institute of Regenerative Medicine, which is really helping to traverse that valley of death. So we've been able to take advantage of their funding mechanisms to be able to fund our phase 2 clinical trial, which led to our partnership. So what does all that mean? What it means is that cell therapy for heart disease is going to be, I believe, the future of therapy for the heart, for heart disease. It's just a question of being able to get through those hurdles, raise the appropriate funds, and be able to separate out the wheat from the chaff. And I think as a funder, if you're one of those people in this audience who potentially could make an investment in a company, it's really tough to figure out who the winners are, because I could stand up here and give a passionate talk about why CDCs, our cells, repair the heart, improve heart function, are manufactured easier, and probably the best therapeutic. But 7 of my competitors would be lining up behind me to tell you why theirs was better. And so I think what we have to do is keep hoping for the field to move forward, to keep a positive energy and support all of our collaborators and even our competitors, because ultimately a win for somebody is a win for all of us. We need to show that cell therapy for heart disease will work. How do we do that? We do well-powered, well-funded clinical trials, which are very, very hard to do because it's hard to get that money. And that's one of the reasons why we're so lucky to have the support of CIRM behind us. They funded a 300-patient, randomized, placebo-controlled, double-blind trial, one of the largest phase 2 trials that will be done in the field, and will allow us to really get to some good answers. And then we have J&J, who's come in to do not only an option to potentially take our cells in, but also a manufacturing collaboration, so that when ultimately we're ready to go to pivotal trials into the market, we'll have a commercially viable product. So for us, that's exciting. I guess I want to close with saying the thing that impacted me the most today was when I was able to sit into one of the, I think he said, non-biologic, non-medical sessions, where I heard about the drive of the public to move therapies from bench to bedside. And the AIDS initiative, which there's many documentaries out there now, that this relatively small group of iconoclastic gay men were able to rally an entire country around themselves to have a therapy developed and a vaccines developed and all of the therapies that they can derive, because they had the political power behind them. They were able to marshal that. And then you see that today, I don't know any of you who have read CNN, the 2 Ebola patients were released for memory, and they got this drug, I think it was called ZMapp is what it's called, and it had not barely made it out of mice. And what do you think the likelihood that company would have survived is without those 2 patients and their life and death need? Is very unlikely that we'd ever have heard about them. But now that company now has a chance to really be able to take potentially something that could be life-saving for many patients with Ebola and move that program forward. So I think the end result here is you all are interested in moving the field forward. Let's also be rebels, and let's also get out there and fight for what we believe will make the world a better place, and our children can live longer to that 122 years I heard earlier today. Thank you very much for your time. I appreciate it.
Thank you, Linda. Eric.
So I realize I'm the last person between you and questions, more importantly dinner and the comedy show, so I won't even use a timer. But I just wanted to really bring up three general points. And the first one, and I understand, unfortunately I had to miss it, but it may have come up this morning already, is that with regard to Alzheimer's disease, despite what you may read in the media sometimes, it's not all so much gloom and doom. I think we're actually at a very promising point in research for Alzheimer's disease. We're not there yet, but I think we're getting very, very close. And that's for a number of different reasons. One of which is related to the ADNI study. That's the Alzheimer's Disease Neuroimaging Initiative, is a public-private partnership. We'll come back to that. Funded by the NIA along with a number of pharma companies and not-for-profits. It's taught us a ton about the pathology of the disease. 10 years ago, it wasn't widely appreciated that your plaques developed 15 years before you developed symptoms. That's a huge window of opportunity to actually have an effect on either delaying or possibly even preventing the onset of the disease. That's information that's only come across in the last 10 years or so. As Rochelle mentioned, there's actually some fairly promising clinical data out there right now. Some of it is on our Lilly drug, solanezumab, but also crenezumab. We think that we're starting to be able to have some treatments. It's not, these first treatments won't cure the disease, but it'll tell us we're on the right track. It'll be a toehold to say, here's where we start, and perhaps this is just the beginning of the beginning, but at least it's the beginning. So the first point is just, despite what you read or what you see on the news, it's not so much gloom and doom. The second point is related, and it gets to a topic that's a little bit esoteric in terms of drug development. And I understand there was a comment I did hear this morning, or it was mentioned that the only way you can get funded now is to do translational research. And I don't know if that's quite the case, but we've actually learned a lot about translational research in terms of Alzheimer's disease in particular in the last 10 years. And so prior to that time, you did some animal studies, you got some PK, you got into the clinic, looked pretty safe, you have your PK and you're off to the races. What we've found is it's very important to show that you've engaged the target in the brain before you go into these very large, long, expensive phase 3 studies. And so now of the molecules that are currently in phase 3, again, we have solanezumab, that's the one that I work with, and we think we have good evidence of target engagement. But Merck has a BACE inhibitor in phase 3. I think there's pretty good evidence of target engagement for the BACE inhibitor. Roche has another monoclonal, gantenerumab. I think there's pretty good evidence of target engagement for that molecule. So again, this kind of gets back to the point a little bit about it's not so much gloom and doom, but I think having learned that you have to do your homework, you have to show that target engagement step, has really been beneficial for the field. And Rochelle may cringe when I say this, but my prediction is of those 3 molecules that are in phase 3 right now, there's a 75% chance that at least one of those is going to show a clear biological effect. Now, it's no guarantee of regulatory approvals and whatnot, but I really think we're on the right track. And so even though it's kind of nuts and bolts, maybe a little bit boring drug development, I think we're a lot further along now than we were 10 years ago. So finally, the third thing I want to point out, and I think I'm really going to be preaching to the converted here, is this is just a huge public health issue. So if you just look at aging broadly, as you know, in the United States alone, 10,000 people, 10,000 baby boomers a day become age 65. Right now in the United States, there's over 5 million people with Alzheimer's disease, over 30 million people worldwide. Those numbers will triple by 2050, it's estimated, if there's not something done, where we can get earlier in the disease process and really slow down the progression of the disease, or perhaps even prevent or delay the onset of the disease itself. And so given the fact that this is such a public health issue, I think as we've already heard a little bit about, different funding sources, public-private partnerships, I think certainly as a country in the United States, but we really need to get together and think about ways that we can attack the problem more carefully. The biggest rate-limiting step right now to better therapies for Alzheimer's disease is recruitment in clinical trials. Our Lilly CEO mentioned to me once of our R&D budget, it's a lot of D and not so much R. In other words, the basic science guys have gotten really good at getting into the lab, doing the right experiments. It's all robotics. It's all very efficient. Where you really slow down is to get into clinical trials. There's now what's called NAPA, the National Alzheimer's Plan Act. Congress passed this act a couple years ago, included all kinds of people, a lot of bells and whistles, and included everything except for money. So there's actually no funding behind the act. But that and some funding is what we need. And so what we really need to get these treatments to people more quickly is a better infrastructure for doing clinical trials and a better appreciation among people in general about how important it is to participate in clinical trials. So we need more sites, but we need better participation. I think right now oncology is probably the best example. If I get a diagnosis, I go into a trial, and the numbers aren't even all that good for oncology. And I'll just close by saying, and this may be a little counterintuitive, but if you really want to make the point about why it's important to participate in clinical trials, you really want to point out that there's study after study that shows that people that go into trials do better than people who don't go into trials, and that's for a lot of reasons. But one of the best scenarios is when you think about going into a clinical trial is not necessarily that you want to get the drug, because especially in a phase 2 trial, most of the drugs actually don't work, and every drug has side effects. What you really want to do is go into a trial and get placebo, which is a little counterintuitive, and actually that may not be exactly the way most people think about it all the time, but I think the point is you're better off being in a trial than not being in a trial. And whether or not you want to be on drug or you want to be on placebo, that's kind of an individual thought. But what we really have to do is change the mindset for people participating in trials and then to build the infrastructure to get those trials done. So with that, thanks very much.
Awesome. Thank you, Eric. Thank you all of you for summarizing so well the work that you've been doing and that you presented earlier. So, as I mentioned earlier, what I'm going to do is spend, what have we got now? I'm going to spend maybe 15 minutes going through a few topics that I feel are particularly relevant, especially to the combination of expertise that we have here on stage. And then I'm going to throw it open to you guys to ask whatever questions you feel like. So I'm going to start with something that specifically focuses on the pair of problems that have been addressed in the 2 biomedical tracks today, namely Alzheimer's and cardiovascular disease. One thing that's always struck me about these 2 things is that they have one thing really in common, namely that they're both very complex phenomena. But they have one thing that really takes them apart at the level of how we normally think and talk about them, namely that Alzheimer's is a bunch of phenomena that we describe as a single disease, tangles plus plaques plus cell loss and synaptic atrophy and so on, whereas lots of different things go wrong in the heart and we give them different names, like atherosclerosis and arteriosclerosis and amyloidosis. And it often seems to me that this is kind of a bad thing, that we ought to be kind of having a holistic, well, let's say an intermediate attitude where we recognize, of course, the panoply, the variety of phenomena that go on in each of these problems, whether it's in the brain or in the heart, but that we also recognize the interactions between them. I wonder whether any of you have anything to say about that, about whether it could be, whether the fields might actually learn from each other in this regard. For example, I'm thinking that in the Alzheimer's world, there was perhaps too much surprise or too much pessimism arising from the results of a trial that just addressed one aspect of Alzheimer's, and so on and so forth. Any comments on any of that?
Well, just 2 thoughts, okay? One is that there's the disease Alzheimer's, once you're symptomatic, and I would say symptomatic with MCI, mild cognitive impairment, defined rigorously, you're a standard deviation and a half away from the mean for your age and education on something important, like memory, okay? So that's Alzheimer's disease. There's a differentiation between that and the pathways you take in the asymptomatic phase. And it may be that there's an endophenotype of Alzheimer's disease that has great overlap with the endophenotype of cardiovascular disease. So I think we have to put a diachronic dimension on this as well as a synchronic dimension, as well as a cross-sectional view. And I think my answers will be different depending on whether we're talking about the unfolding over time or the moment of disease.
Thank you. Yeah, that actually speaks to another of the questions I'm going to ask later on. Anyone else want to say anything on this one?
Go ahead.
Well, okay. So I was just going to with the analogies in terms of cardiology, actually that's come up with us quite a bit. One of our senior VPs is a Harvard-trained cardiologist, and he sort of appreciated the fact that plaques are bad on your teeth, and they're bad in your arteries, and they're bad in your brain. And I think there really are some analogies where you can have asymptomatic coronary artery disease, it's an accident waiting to happen, in the same way that you can have amyloid plaques in your brain but be cognitively normal. And all the data now are showing that that's not going to continue to be the case. So there are a number of analogies. Now, they're not perfect analogies, though. I think cardiac events, maybe with the exception of congestive heart failure, but a lot of those are acute events. So I'm always kind of jealous that, well, do you have an MI? Are you dead? Those are kind of, acute things that are easy to measure. Have you moved from MCI to mild Alzheimer's dementia? That's a really very fuzzy line. So I think there are some analogies that work, and then there are some differences.
Yeah, I have to agree, and I think that one of the things that's traditional across medicine in general, but specifically cardiovascular disease and Alzheimer's disease share, is nobody's really very interested in prevention, even though we kind of talk a lot about it. And go try and fund a clinical trial that focuses on even early stages of disease. And patients don't want to come into that trial. In fact, the clinical trial we're doing now called All-Star, we're focusing on, and for those of you that didn't hear my talk earlier, I go through a nice diagram where I show adverse remodeling is what happens to your heart after an injury. So essentially you have an injury to your heart. It's like an injury to any other muscle. The only difference is the heart's beating 72 times a minute. In response to that injury, the heart changes its dimensions. One of the most beautiful things I think about the heart is that it's a physical organ. It's dependent on physical and chemical properties to function. As the dimension changes, you lead to a paradigm called heart failure, which is the inability of the heart to meet the needs of the circulation. 5-year mortality with heart failure is 50%, and it's an $8 billion a year cost to our healthcare system, so it's a huge problem. And the clinical trial that we're working on now, we're addressing adverse remodeling, which is that's the change in shape of the heart. It's actually where you can prevent that progression. Those trials are hard to recruit. You have patients saying, oh, I'm fine. I have a heart attack. I got my stent. I'm good to go home. And so, it's really hard to get people as patients engaged, and it's also really difficult for the general healthcare providers to care about it. So it's interesting. You hear the same thing. And I think everybody, as afraid as many people are of heart disease, I have to say, I think the most fearful disease of anybody that ages is Alzheimer's. And it's because I think it's insidious. You don't know that it's coming. And so even with something like that, you still have a situation where you're, unable to address it at the earlier stages, or at least until now.
Well, we do have some we're actually recruiting for 2 prevention trials right now. One with pioglitazone and one with the A4 study with solanezumab. The difference is people are touched by Alzheimer's disease even if they don't have it. So many people have an affected family member. 1 out of 3 people knows somebody with the disease. We're actually getting quite a response to people who want to be in the prevention trial.
How do you identify the patients for that?
Right now, we're just mailing out cards by zip code of people who, by proximity, by demographics, are likely to have over age 65 individuals, and mailing out cards, and they're calling in.
Wow, that's fabulous.
So on the basis of what you guys have said over the past couple of minutes, I'm actually going to reorder my questions, because you've said so much about the dichotomy between prevention and treatment that I think it's probably appropriate to ask this question next. In the commercial world, where, well, 2 of you, Dave, of course, it's, as you said, really tricky to do prevention work. Of course, you're doing your best to, but the real challenge is, I've always felt that a lot of that challenge comes from the source, namely the general public, who are ultimately a bit scared of submitting to any kind of medicine when they're not yet sick. They just don't see the cost-benefit ratio appropriately, I think. But the dichotomy that I wanted to ask you all for your views on, and we'll come back to the audience on this one later on, I think, is between how the medical and medical research fields see this problem and find this problem, namely that it's hard to be preventative enough, versus the way that the biogerontology and basic research community addresses it. The particular example that I always highlight here, which will be familiar to those of you from the biology of aging field, is that a few years ago there was this extraordinary result where a particular drug, rapamycin, was given to mice at middle age, 20 months of age, and it had a very significant effect. It had an effect that was pretty much the same as what you get if you use calorie restriction, obviously a very well-studied intervention, starting from the same age. Fantastic result, far better than anyone had ever achieved with any kind of pharmacological intervention at that kind of age. But what is often swept under the carpet is two things. Number one, that experiment was only done by accident. It was done because the formulation of the actual pellets of food somehow underwent some technical delays, and such that the cohort of mice which were intended to be fed this rapamycin starting at a much earlier age only started getting it then. They thought, well, screw it, we'll do the experiment anyway. Okay. The second thing, which is perhaps even more mind-blowing, is that after this result came out, nothing changed. Still, even today, with just one important but nevertheless small exception, namely a study being done at the Buck Institute for Aging Research, there is still almost no late-onset work being done looking at pharmacological or indeed genetic interventions that would significantly postpone the ill health of old age in model organisms such as mice. And I feel that this is ultimately because there's a lower hit rate. You're going to get negative results more often, and it's driven by the publish or perish tyranny that exists within academia. So what I'd like your thoughts on is whether you see any way out of this, any way to kind of reconcile the over-bias in favor of early onset work in academia versus the over-bias against late onset treatment in industry in general.
Well, I could pick up on a little bit of that thread, but I guess I'd like to take one further step back and look from sort of the 30,000-foot view and look at the healthcare system in general, right? And so we don't treat health in this country, right? You go to the doctor when you're sick, so we treat disease. Mm-hmm. And so I think we really need to, there need to be incentives in place to treat health and preventive measures. And so people should really be rewarded for taking care of themselves. So if you don't smoke, maybe you should be rewarded. If you have a good diet, if you exercise, there should be systems in place to encourage that behavior as opposed to just when you get sick going to the doctor, and then that's such a huge burden on society. So I think we need to really shift the way we think about healthcare in this country, and preventive medicine will follow that.
And how do you think we would do that?
Well, that's a good question. So I think it starts with insurers and trying to incentivize good behavior. So whether you have a reduced insurance premium if you go to the gym, or you don't smoke, or you have a good diet, I think it really comes down to cost.
Well, we have people in the audience who are pretty familiar, more familiar, I think, than anyone else on the stage with the insurance and pensions industry, so we'll hear from them later on, I'm quite sure, on this point. So please hold that thought, everyone who feels you have something to say. Meanwhile, there's one other thing I wanted to bring up on the commercial side, which is the incentive to do long-term thinking. One thing that happened quite recently was that about a couple of years ago, some very preliminary work came out showing that one aspect of aging, namely the accumulation of senescent cells, was, first of all, rather bad for you, and second, in mice, and second of all, if one went in late and removed these cells, there was good health benefit. And this, even though, as I say, it was very preliminary work in all manner of ways, which I could enumerate, it actually got funded. They actually managed to get good capital for a startup company that is now moving forward trying to emulate this pharmacologically. This seems to me to be a wonderful piece of good news, and I think it arises from the fact that people are beginning to recognize the enormous potential, commercial potential in the end, for therapies that really comprehensively postpone age-related ill health by a lot. That will be the biggest industry ever by a very large distance. But it's still happening too slowly. I'm wondering whether any of you, especially those of you with expertise on the private sector, have ideas about how we might accelerate this kind of sense of proportion, this appreciation of just how appropriate it is to take a longer view, a more high-risk, high-gain view commercially in moving forward with what we might call anti-aging medicine that really works.
So maybe I can try and tackle that one a little bit. So I think you're right. I think the current model is, from a pharma standpoint, that if you get too early in the target identification process, you can't do it from a pharma standpoint. The way, whether it's due to patent law or whatever it is, it's too long of a time period. So the current model is that you work with you follow a lot of academic work, and when it gets to a certain point, and a lot of times you have, sometimes it's a purely academic lab, sometimes it's a little biotech, somebody come to a big pharma company, well, there's a certain criteria where, do you have enough data or not quite, go do some more experiments. It's a little bit like trying to finish your PhD or something, I guess. But anyway, do you need some more data? So that's the current model. But I think that, and I'm more familiar with this actually from the clinical side, but there needs to be a lot more de-risking, and there needs to be a lot more done in the way of public-private partnerships. So in other words, and I know I get actually involved in a lot of different discussions of various public-private partnerships, and some of those do go back actually very early in the discovery process to try and look at new targets and look at new potential targets. But it's very risky, and I think to find ways of sharing risk and de-risking things is one of the ways that you could move that along.
Go ahead. I was going to say, just picking up on that thread, I mean, I think the public-private partnership is essential, and that's really what CIRM is here to do, is help de-risk novel technologies and novel therapies to get them to a point where industry and the private sector can then pick them up and move them forward and actually commercialize the products and deliver them to patients. CIRM's not going to end up doing that at the end of the day, but if we can use our capital in a non-dilutive sense for companies like Capricor or Sangamo or any number of our grantees to attract private sector dollars to then move these projects forward, I think that's a good private-public partnership. And I think there are examples of companies, big pharma, getting involved earlier than they may otherwise would if CIRM dollars weren't there. So I think to pick up on your point, it's essential to drive this research forward.
I'd like to elaborate on that. I think something that you said earlier when you were giving your comments about identifying the right biologic target, and that the clinical trials are far more successful once you know your biomarker that you're going after, that you have some biologic activity. And I can say that when we founded Capricor, I looked at the graveyard of biotechnology, and I decided that I wanted to do it very differently, because the R, and he spoke of little r and big D, and even from the baby company, that's exactly my philosophy. We are tiny r, big D, because what we want to do is we want to take something that's been well hashed out on the academic side. And if you actually look at our company, we've brought in $12 million of equity investments into phase 2 for our product, and we actually have 3 products now, and $39.5 million in non-dilutive capital, largely through grants and a partnership. And we also then access the academic labs that we partner with, the KOLs, to the tune of, we've estimated, in the $40 to $50 million range. And this was to get a product into the clinic. And so we have to, as a mandate, strengthen those relationships between the private and the public, and let the academics drive the agenda into the early stages. And one could argue that the early stage really doesn't end until it's in the clinic and in a person, because I think we all know that if we could cure diseases in mice, we would all be living to 200 years old as Methuselah.
So that's actually a great lead into the last question I wanted to ask before I open it up, which is actually, I guess, mostly directed to Neil, but certainly all of you, I welcome your comments, which is essentially what I would say is the valley of death between R and D. So in the commercial sector, for the reasons that you guys have very much laid out, it's always going to be much more D than R, right? But in academia, especially, well, at least in the bulk of government-funded academia, there seems, within the biology of aging at least, to be an over-bias in the opposite direction, where it's all R and absolutely no D whatsoever. And between the two, you've got what you might call early-stage development, stuff that is the proof of concept work, it needs to be done before industry can really pick up, but still is very much goal-directed as opposed to curiosity-driven. it's the kind of reason why SENS Research Foundation exists, to be perfectly honest, as a non-profit, that we bring in funding from philanthropic sources predominantly to do work that takes basic science knowledge forward and far enough so that it's within the visibility of the private sector. As my CEO, Mike Cope, often mentions, our ultimate goal is to try to make ourselves obsolete by kickstarting a biotechnology industry, and we use the word industry very advisedly. So, Neil, I wonder whether you have any view as to the role that an organization like CERN, which essentially, is set up, if you like, to complement the federal government and such like, can play to fill that void.
Sure. No, I think that's an important question. And, I think from particularly the perspective of cell therapies, I think it's hard to separate the R from the D. I think in many ways, the product is the process, as people like to say in the cell therapy world. And so it needs to be an iterative feedback loop. And I think in terms of de-risking projects, it's not only capital that does that, but you have to have standardization. You need to have a solid CMC process. You need to make it manufacturable and scalable. I think all of these things, and it has to be a feedback loop between development, process development, and research. And the biology and the engineering have to go together. And I think CERN plays a role to fill, if you want to call it the valley of death, although I don't like that term, the translational gap. CERN provides funding to help sort of bridge that gap. And whether it's driving towards proof of concept, which is what we all want to do, but part of it has to be providing funding for that feedback loop between the R and the D and the process development and the manufacturing. So I think CERN plays an essential role there.
Great.
Just a quick comment. We need to change the rhetoric. I mean, the more we talk about valleys of death, the more we talk about failure rates, then we get, as we were talking about, to recruitment, and we want to bring people into prevention studies and they're not sick, or they don't feel sick. We want to bring people into mild stage clinical trials where the uptake rate used to be 90% and it's now 30%. Of any interest at all in being in a trial because they have another therapy. We have to change the rhetoric. And everybody does this in their grants. The first line is there's no treatment for this incurable progressive disease, fund me. That ultimately is self-defeating.
Why do you say it's self-defeating?
It's self-defeating because then people get the message that it's no good, okay?
Yeah, it's negative.
It's the rhetoric around it, the reporters who report on it, the commentators who comment. Everybody will pick up that rhetoric and feed it back to the public who's going to say, well, I'm not going to do it.
So thank you so much. We have 15 minutes now, and I will be outraged if these guys haven't annoyed you enough to ask a lot of questions. So please, anyone who has anything to say, go to the microphone. On my right, first of all.
This is I guess it's for everybody. We now have 3 new potential diagnostic tests for Alzheimer's. One that came out of UK, I think is that the one with the 10 proteins? And within a year, they'll be able to give you some idea of MCI or Alzheimer's. And then there's one from Georgetown that is going to do the same thing, but it's using lipids, I believe. And then there's another one. I got to read this off my text messages. It is retinal eye scan by neuroimaging. And that one, Neurovision Imaging Corporation. And that one is supposedly in stage 2 or so of clinical trials and is going to predict Alzheimer's up to 20 years in advance. What do you guys think this is going to do to the public and to clinical trials and for Alzheimer's?
Go for it.
Start with that. Well, first of all, there are 100 potential diagnostic tests out there. Those are the ones that were picked up by the media most recently and circulated. And the part that gets missing in your description and in their descriptions is what exactly was being tested. So in the lipid profile, what exactly was being tested was the ability to predict the likelihood of going from mild cognitive impairment to Alzheimer's disease, which is also Alzheimer's disease. So that's actually possibly a biomarker of progression. at a certain stage. That's not the same thing as a diagnostic test. So what we hear is an overgeneralization. There are scores of profiles, whether they're lipidomic, proteomic, there's scores of profiles out there that someone has put forward that is not refined, not proven, and is not even under consideration as a diagnostic test yet. It may be a differentiator between cases and controls, or it may tell which MCI subjects will get worse sooner. So we're not on the threshold of a diagnostic test. Any one of those could become one, but we're not on the threshold of that. If we get something that's a diagnostic test, we'll then have to learn whether it diagnoses all forms of Alzheimer's disease or just people who come to have Alzheimer's for a particular reason. But I think it would be an aid if one of these things is successful, but I do want to caution you against believing that each and every one of these headlines is really a test that's just around the corner.
Yeah, especially based on small early studies. So let me just build on that a little bit and just full disclosure. So Lilly has a PET ligand called amyvid, which is a diagnostic test. You get a PET scan and you see whether you have plaques. It was actually developed by a small biotech called Avid. We used it in our studies, and it's like the old commercial. We like the company so much we bought it. So, but there are 2 other PET ligands that are now FDA approved, and certainly not going to get into a debate about which one's better. But the bottom line is each one of those things will tell you if you have plaque in your brain. Now, what people are really looking for is something that is cheaper, doesn't require radiation, a better way to do it. And so there are a lot of different people who've looked for a lot of blood tests along the way. And so in order, especially not to be too negative about this, I think someday somebody's going to get lucky, and it's going to take some luck, because with every one of those studies, without going into the details, there's a better chance that it's a false positive than a true positive. But eventually, somebody's going to get lucky with one of those. And the one other point that I did want to make, because there may be people in the room who are involved with this, is the people who are working on blood tests right now seem to think that it has to be perfect. It has to have really high sensitivity and specificity. I don't think that's the case for a blood test that's cheap and easy to do. You want sensitivity, but you don't need so much specificity, because all you're going to do is determine who gets the PET scan, which is the real definitive test for whether you have plaques. So if anybody's working on a blood test, don't try and make it perfect. Just try and make it very sensitive.
Ben.
Yes, as a non-MD, I may be totally off base, but my feeling is that in terms of preventive medicine, there are 2 things that we need to look for. One is some kind of an intervention which will prevent the ultimate long-term disease. And the other one is immediate, relatively fast feedback to the general public, to the patients. And the only exception to that rule is smoking, okay, where the campaign has succeeded tremendously without having immediate feedback. Okay, so having an accurate diagnostic test which will predict that you'll have Alzheimer's 15 years or 20 years from now is basically useless unless you have some way of intervening. A good example to that is coronary heart disease. Okay, we have a cholesterol test, we have blood pressure, we have statins, we have antihypertensive drugs. So Joe Schmo, pardon the expression, goes to a doctor who tells him, look, listen, if you continue living the way you are, you will have a fatal heart attack 30 years from now. That's useless. But if he goes to the doctor tells him, look, you have to lower your cholesterol below 200, your blood pressure is too high, here's some medication, do it. That works. Okay, so I think there needs to be a focus on, in addition to long-term diagnostics, an accurate diagnostic needs to be a focus on finding feedback mechanism, finding interventions which will prevent the problems from the future.
So I have this premonition that
it's a very high order, of course.
I have this premonition that Jerry's going to be in soon and get me to speed things up. So if we could have quick questions and quick answers from now on, please. And I'll be able to take all the people who are standing up right now, but nobody else. Thanks. Go ahead, please. Anyone got comments on what Ben just said?
Well, you seem to be saying that you not only need the intervention, but you need a biomarker or something more proximate to say that it's working. I agree with all of that. I don't want to discourage the field, but I do want to say if we get a vaccine for Alzheimer's disease, the government's going to have to require it in order for people to use it. Because people don't go in and do these things. You know, they don't even vaccinate their children necessarily if they didn't have to do it for school. So, you know, it's going to be a public health issue and not just a logic or common sense or medical issue.
I guess let's go back to something that was said earlier about how incentives, financial incentives can perhaps work here. Maybe it wouldn't need to be required so if one could get, for example, health insurance companies to offer very substantial incentives in terms of premiums.
Right. I was trying to be quick.
Right.
I actually disagree. I think if it was a vaccine, you know, think of immunization programs. They're very successful. I think of
because they're required. They're required. And honestly, you know, what people want is something they can eat. Okay? Universally. I want something that I can eat that is going to prevent X, Y, or Z.
And the lipid-lowering drugs in cardiovascular disease, he mentioned those. You know, the statins have been enormously successful in reducing cardiovascular risk.
Yeah, but what's the compliance rates?
Terrible.
That I don't know.
Terrible.
They're low.
Next.
I reconnect a little bit on the preclinical versus clinical problem that you mentioned, Eric, a little bit about the different pace between the two processes because preclinical is much faster. But it's also much cheaper than clinical trials. So what is, since you are all expert on cardiovascular or Alzheimer, what is considering the state of the art of potential therapies, now, do we need more money into the trials or do we need more money to improve the therapies at the basic level?
Well, by personal opinion, that's a great question. I say the money needs to be there to build the infrastructure to do better, faster clinical trials.
Well, I actually think, I thought about it for a minute, I actually think that if we could do better at the basic level in identifying those relevant targets, identifying the biomarkers that they express, and then their downstream clinical consequences, we could do smaller, more effective clinical trials. Yeah. So maybe, you know, sort of pipelining it.
Yeah, and there's going to be some very, well, actually today as well, but throughout the meeting we've got discussions in the non-biomedical tracks about how to improve the efficiency and the informativeness of clinical trials. I think there's a lot of low-hanging fruit there.
Yeah, I would agree with that. I mean, I think it's twofold. I think the underlying biology drives the development. So the more you understand the underlying biology, the more efficient you'll be able to make clinical trials, the more targeted you'll be able to make clinical trials. So I know CERN, for instance, we're very focused on funding translational medicine and clinical trials, but we still fund basic biology because you can't leave that behind. So.
Very good. Doug.
On the theme of preclinical, trying to save money on clinical trials, are there any mouse models that you believe in? For dementia.
I'm not touching that one.
Yeah.
I think that they inform, you know, they inform. There's no mouse model that I think is better than another, or there's no mouse model that would convince me that one drug is better than another. I think there are mouse models in which it's easier to move amyloid. So if you have something where what you really want to know is does it move amyloid, you use that mouse. It's probably best for you to use another one too.
But there's some basis for moving on to human. And would you consider mouse to be the primary gating factor after you've identified a much larger number of upstream targets? Or
I think that one way to think about the mouse models for Alzheimer's is there's models of amyloid deposition and there's models of tau deposition. There's no model of Alzheimer's disease.
Yeah, even the triple transgenics are basically just putting the three things together in isolation rather than putting them together in the way that they actually interact in the human. Right.
Okay.
Richard.
Yeah, so I wanted to have a question of a rather different order about models. And it may turn out to be not answerable in the minus 30 seconds that you've got left. But anyway, let me ask it. So we all know about the model of innovation that this whole industry has been based on, right? A welter of academic publications followed up by biotech companies racing for the funding. Then they go through the valley of what I would like to suggest is difficulty rather than death. But they do go through it, and that's where folks like SENS and and CRM are useful. Then the big companies come in, and then the regulators, then the reimbursement agencies, and so on. I'm increasingly wondering whether that whole ecosystem model, that linearity of it, actually can grapple with the kind of big issues that you're throwing out there. The patients don't get involved until clinical trial recruitment or taking it at the end. Government doesn't get involved until they have to pay for it right at the end, unless they put money into something like CERN. Is there a different ecosystem struggling to get out to tackle the kind of problems that we're talking about?
That's a great question. I have no idea what the answer is.
That's a session for next year.
Well, you can raise the advocacy group initiated research. I mean, the families with, afflicted with rare genetic diseases have really developed a model for getting involved in what basic science gets done, what gets funded, what gets moved forward into populations, and when. And I think, you raised, Linda, the Ebola example. There's a very articulate scientist who's been on the news a lot talking about barely holding himself back to talk about how long it usually takes to get something into humans. And so, things are getting into humans faster. We may have a model, and I was thinking about that today, where we start giving little groups of people all over the place, not carefully selected, all sorts of drugs very early in disease, early in drug development. And then it'll be like the age of Pasteur, then we'll decide whether to take those things forward. It seems like we're moving a little bit back toward that.
I hope so.
I'd just like to mention that Richard is somewhat being British and modest because he's actually fairly involved in exploration of areas like adaptive licensing and such like to speed these things up. So this will really be the last question. Go ahead, please.
My point, or it's a question, and unfortunately I can't formulate it really clearly because I don't think it is clear to me, relates to the model issue. Most of the discussion I've heard regarding the Alzheimer's track today has been in the paradigm of the medical model and the pharmaceutical model extended backwards in time to an earlier stage, saying, can we take this model earlier? Can we fund more clinical trials? Can we investigate interventions, which are basically pharmaceutical interventions, earlier? On the other hand, I submit that some of the greatest achievements in health and longevity that we've experienced as a society have come from a different model, more of a public health perspective. I'm thinking of things like vaccinations. I'm thinking of things like the cleaner air, cleaner water. I'm thinking of things like resisting smoking, of campaigns, which was a very simple message: stop smoking. Now, it could be, but we don't seem to have a concerted approach to aging within this model. It seems to me we need to take on aging as a public health generic issue. And this may open up new dialogues and new discussions. For example, I'm very aware of the siloization of research related to aging today. It's incredible. And we didn't hear anything about metabolics today, and there's a lot of work going on in molecular metabolics now, a tremendous amount. And some of that may well apply. For example, I heard a comment earlier today that one of the origins of the tau stage of Alzheimer's disease, the tau tangled stage, one of the creating factors may be a failure in the lysosomal system and a failure of clearance of dead mitochondria or dysfunctional mitochondria. Well, there's a tremendous amount of research going on over here that we didn't hear about this morning related to that. And so it's a question of how can we connect these things up And how can we bring this out as a public health issue?
So I didn't plant that question, but it's one of the best advertisements for Sense Research Foundation that I've heard today. It's exactly the kind of thinking that has led to the kind of cross-disciplinary work and indeed cross-disciplinary conferences and sessions that we have been pursuing and promoting for the past many years. And I assure you that this is something that certainly I and those that I speak with understand very well. I think, however, what it really often comes down to, from my point of view at least, is a mindset problem of, as you say, over-balkanization, of thinking of different diseases as separate, but especially of thinking of these diseases as separate from this other nebulous thing that people have in their minds called aging itself. So that was what I tried to focus on this morning before lunch, of course, to emphasize how the erection of a completely artificial Chinese wall between the things that we call diseases and the things that we don't in old age is massively counterproductive. Other comments?
Yeah. One, that, again, it just occurred to me today from being here. You know, aging is a process, an undeniable one, and the things that you said are very applicable, and we didn't go over it, but there are actually a family of interventions that are related to that kind of thinking. Most of the early stage intervention trials in Europe are actually multimodal interventions of diet, exercise, socialization, and other factors. But the comment for me today, the take home for me, aging is a process that's undeniable. Age is a social construction. It's like the difference between sex and gender. And so I think when we think about age or our age or someone's age, I think I'm going to start keeping that in mind because there's room for intervention around that too.
All right, well,
really,
oh, sorry, go ahead.
I'm sorry, I just want to say I applaud that thinking because I think it's very important.
Yeah, I think I completely agree too. All right, well, thank you. I would like, perhaps everyone could thank our speakers in the usual way.